The patch clamp technique is a widely used method for analyzing the electrical properties of ion channels and the action potential of excitable cells. It is like a molecular superpower that lets scientists and researchers understand how ions flow in and out of cells, which is key to understanding how nerves and muscles work. But mastering this technique is not for the faint of heart. It takes a lot of practice and precision to get it right. But don't worry, we're here to help. In this video, we'll walk you through the ins and outs of the patch clamp technique step by step. This means that you can study very fast electrical events, such as action potentials and synaptic currents. They won the Nobel Prize for Physiology or Medicine in 1991 for this discovery. So don't get discouraged if you don't get it right the first time. With a little practice, you'll be a patch clamp pro in no time. And who knows, you might even discover your own patch clamp magic. By the end of this video, you'll have a good understanding of how to use this powerful tool to study ion channels. So let's dive in. First, you need to prepare your equipment. That means getting your electrodes, amplifiers, and software ready to go. The patch clamp technique can be used with both single and multi-electrode setups, each with its own advantages and disadvantages. For cell-attached recordings, you'll want to find a spot on the cell membrane that has a high density of ion channels. For whole cell recordings, you'll want to find a spot that's easy to break into. Once you've found the right spot, it's time to make your second critical decision. Are you going to do voltage or current clamp? Voltage clamp is a technique used to study how cells respond to changes in membrane potential. Current clamp, on the other hand, is a technique used to study how cells generate and propagate action potentials. Once you have your equipment ready, it's time to make your first critical decision. Are you going to do cell-attached or whole cell patch clamping? Cell-attached patch clamping allows you to study the electrical properties of a cell without breaking the cell membrane. This can be useful for studying cells that are difficult to break open, or for studying cells in their native environment. Whole cell patch clamping, on the other hand, involves breaking the cell membrane and recording the electrical activity of the entire cell. This can be useful for studying cells that are easier to break open, or for studying cells in a more controlled environment. Next, you need to find the right spot to record from. Finally, it's time to start recording. With the patch clamp technique, you can record the electrical activity of a cell with very high temporal resolution. Patch clamp magic was first discovered by Erwin Neyer and Bert Sackman in 1976. We hope you enjoyed this whirlwind tour of the patch clamp technique. Remember, practice makes perfect.